Hello everyone, welcome to Tawhid Foundation for Allied Healthcare Sciences, Bangalore. Today's class is about MIC and MBC. MIC, which is nothing but minimum inhibitory concentration, and MBC, minimum bactericidal concentration for BSc MLT second year students. So, why it is necessary to study MIC and MBC? The rapid emergence and spread of antibiotic resistance among pathogenic bacteria pose a significant threat to global health. In this context, MIC and MBC are invaluable tools that allow clinicians, researchers, and regulatory agencies to assess the potency and real-life effectiveness of antibiotics. MIC is defined as the lowest concentration of an antimicrobial agent needed to inhibit visible growth of a microorganism after overnight incubation. Determining the MIC helps clinicians select an antibiotic and dosing regimen most likely to achieve therapeutic success while minimizing toxicity. Because each microorganism, patients, and drug interact differently, so MIC testing brings crucial specificity to individualized antibiotic therapy. Lower MIC values indicate higher effectiveness. Drugs with low MICs can be used at lower doses, reducing side effects and slowing the development of resistant strain. MBC is the lowest concentration of an antimicrobial needed to kill 99.9% of the bacterial population. The distinction between bacteriostatic, which is growth inhibiting, and
Hello,
bactericidal, which
madam.
is killing. Yes, sir, I'll go to next slide. I'm just giving
Yeah,
a
yeah.
short intro.
Right,
Yes, sir. So these growth inhibiting and killing activity has direct clinical implications. So in summary, studying MIC and MBC is fundamental for optimizing treatment choices, improving patient outcomes, reducing adverse side effects, supporting public health interventions, and guiding the future of antibiotic research and policy. So their continued use and understanding are indispensable in the fight against infectious diseases and antimicrobial resistance. So coming to introduction part to antimicrobials, antimicrobials include antibiotics, antifungals, and antiseptics that kill or inhibit microorganisms like bacteria and fungi. So they are indispensable in treating infectious diseases. However, misuse can lead to resistance, making it essential to accurately assess antimicrobial potency. So standardized testing such as MIC and MBC helps in selecting the right drug and dosage to avoid resistance. So, what exactly is MIC? The MIC is defined as the lowest concentration of an antimicrobial agent that prevents visible growth of a microorganism after incubation. These parameter reflects the bacteriostatic activity of the drugs, meaning it stops bacteria from multiplying. So MIC helps clinicians determine whether an antibiotic is effective against a particular pathogen, guiding treatment choices. So these points, what is MIC? It is lowest concentration of antimicrobial that visibly inhibits growth. It will measured in μg per ml or mg per ml, determined in vitro, which is controlled lab condition. It will indicate bacteriostatic activity, and it is essential for choosing effective antimicrobial therapy. Importance of MIC. So, determining MIC is vital to guide appropriate antibiotic use and avoid unnecessary or ineffective treatments. It helps in dosing optimization to minimize side effect and development of resistance. MIC data also plays a role in epidemiological surveillance and aids researchers in screening new antimicrobial compounds. So, authorities like CLSI and EUCAST will set standards for MIC interpretation. Principle behind MIC test. MIC testing is based on exposing bacteria to a gradient of antimicrobial concentrations in vitro. Standardized bacterial suspensions are added to tubes or wells containing serial dilutions of the drug. Following incubation, tubes are examined for bacterial growth, typically visible as turbidity. So the lowest concentration without growth is recorded as the MIC, which is nothing but minimum inhibitory concentration. MIC test procedure part 1. So the first step involving preparing a bacterial suspension matching a standard density, serial two-fold dilutions of the antimicrobial are prepared in broth or agar. Each dilution contains decreasing amount of the drug, allowing identification of its inhibitory concentration. Sterile technique is crucial throughout to avoid contamination. So here you'll prepare bacterial suspension at a standardized density, example 10 to the power 6 CFU per ml, and then prepare serial two-fold dilutions of antimicrobial in broth or agar, and then dispense dilutions into test tubes, sterile test tubes or microplate wells, which is 96 deep well plate, and then maintain sterile techniques throughout the procedure to avoid contamination. Why it is not? So for part 2, next, each dilution is inoculated with the bacterial suspension. The tubes or microplate wells are incubated under optimal temperature conditions, usually around 35 to 37 degrees Celsius for almost 16 to 20 hours of incubation. After incubation, look for turbidity indicating growth. The MIC is the lowest concentration where the broth remains clear, signifying no growth. So after inoculation, inoculate each antimicrobial dilutions with bacteria and then incubate those tubes or wells at 35 to 37 degrees Celsius for up to 20 hours, and then next day observe visible growth, which is nothing but turbidity or pellet formation down the test tubes, and then record minimal concentration with no visible growth. That means it will be transparent or clear test tube. Include positive and negative controls as well. Types of MIC test. Broad dilution is so these are the types of MIC test: broad dilution, agar dilution, e-test, automated system, example Vitek, and choice depend on lab facilities and the purpose of the experiment or test. So broad dilution, serial dilutions and liquid medium. Here, broad dilution is the most common method performed in tubes or microplates. Agar dilution, it involves incorporating antimicrobial agents into agar plates and spotting bacteria. E-test, it uses gradient stripes to determine MIC on agar. Automated systems, instruments provide rapid and standardized MIC measurements. So each method has advantages based on lab capacity and study of that experiment. Reading MIC results. So interpreting MIC involves checking for visible signs of bacterial growth. Clear or transparent broth indicate growth inhibition. The first dilution with no turbidity sets the MIC. Testing is usually duplicated to confirm accuracy. MIC results must be compared to clinical breakpoints to understand their therapeutic significance. So clear broth here, it indicates no visible bacterial growth. That means bacteria is not present in that broth. Turbid broth, bacterial growth. That means bacteria has grown in that test tube. MIC, lowest concentration where broth is clear. It will confirm with repeat testing if it is needed, or if you'll have any doubt the reading is not proper. Compare values with clinical breakpoints for interpretation of results. Factors affecting MIC. Several variables affect MIC accuracy. The bacterial inoculum size must be standardized. Too many or too few cells can skew the results. So medium components, temperature, pH, and incubation times influence bacterial growth and drug activity. So some bacteria in biofilms or with resistant genes may require modified approaches. So attention to all parameters ensures reliable results. So what is the main factors which will affect MIC? It is nothing but size of this bacterial inoculum. It can change the result, actual result. Temperature, pH, and incubation, these three things can influence the growth of bacteria. Composition of growth medium, it will also affect antimicrobial activity. Some bacteria will also form biofilms, altering susceptibility, and precision in dilution preparation is critical. So this image likely shows a series of tubes or wells containing a microbial culture medium with increasing concentrations of an antimicrobial agent. So the tube starts clear or turbid depending on the organisms of growth. The first tube without visible turbidity or growth, it indicates the MIC. So either it can be done in tube or plate method, depends. So this is the plate which will show interpretation of microdilution of MIC results. What is MBC? MBC is nothing but minimum bactericidal concentration. While MIC measures growth inhibition, MBC measures bacterial killing. It is the lowest antimicrobial concentration, reducing viable bacteria by 99.9%. The MBC test complements MIC by confirming whether the bacteria are merely inhibited or fully eradicated. So this distinction is critical in infections where complete bacterial death is necessary. So minimum bactericidal concentration is the lowest concentration of an antimicrobial that will kill around 99.9% of bacteria. It is determined after the MIC test by subculturing onto antimicrobial free agar plates. It will indicate bactericidal, not just bacteriostatic activity. It will be measured in μg per ml or mg per ml like MIC, and it is essential for infections requiring bacterial eradication. Importance of MBC. MBC is important in cases like bloodstream infections or endocarditis, where killing bacteria rather than just stopping growth is essential. It helps clinicians decide if a bactericidal rather than bacteriostatic antibiotic is needed. Additionally, MBC helps pharmaceutical research by assessing the true killing efficacy of new agents. So why it is important MBC? It will confirm the actual killing ability of antimicrobial agent. It will guide therapeutic choices for several infections. Example include endocarditis infection or meningitis, etc. It will help differentiate bacteriostatic as from bactericidal drugs. It is critical for research as well as development of new antibiotic drugs. It will support infection control and sterilization protocol development as well. So principle behind MBC test. After determining MIC, samples from clear tubes, which means no visible growth, are subcultured on antibiotic-free solid medium. The absence or presence of bacterial colonies after incubation indicates bactericidal activity. The lowest drug concentration from which no colonies grow is recorded as the MBC, indicating effective bacterial killing. So once MIC test is done, samples with no visible growth, that means clear test tubes, are plated on drug-free agar, and then viable bacteria will form new colonies indicating survival. The lowest drug concentration yielding 99.9% reduction in bacteria is nothing but minimum bactericidal concentration. MBC must be determined for each microorganism and antimicrobial combinations. These tests will actually kill not just growth inhibition. So it will actually kill the bacteria and not inhibit the growth of an organism. MBC test procedure. Tubes or wells showing no growth at MIC are selected for plating. A small electrode is spread onto agar plates without any antibiotics. Controls include drug-free inoculum, which is growth control, and sterile medium as negative control to validate result. These steps differentiate viable but non-growing bacteria from those effectively killed. So these tubes and wells from MIC test which has no visible growth were then used forward for subculture. That tube will spread aliquot on antimicrobial free nutrient agar plates or any selective media, which will include positive growth and sterility control. Positive growth, it include growth control, drug-free inoculum, and negative control, that means a sterile medium, and then it will be incubated for 24 hours at 35 to 37 degrees Celsius. Sterility technique is very much important here to avoid any further contamination. Next is part 2. So agar plates after incubation at optimal temperature for 24 hours, colony counts are performed, which means 99.9% reduction of CFU compared to control defines the MBC. So repeatability is important for test reliability. This process ensures that the MBC truly represent killing and not just inhibition. So after incubation, colony count colony forming units, which is nothing but CFUs. It is the full form of CFUs on each plate, and then identify which is having lowest antimicrobial concentration less than 0.1%, which is original CFU count, and then it will be documented MBC value and compared it to MIC values. This will repeat test for accuracy and validation process, and then results will be reported with clinical relevance notes. So difference between MIC and MBC. MIC assesses the lowest concentration that prevents bacterial growth, indicating bacteriostatic effects, whereas MBC determines the concentration required to kill bacteria, indicating bactericidal effects. MIC testing is quicker and more commonly performed, while MBC testing is more labor intensive but critical for certain clinical decisions. So here, as you can see, the distinguish between MIC and MBC. So aspect, definitions, purpose, method, result, and clinical use. These are the terms against MIC and MBC. Definition as MIC, it is lowest concentration to inhibit visible bacterial growth. MBC, lowest concentration to kill 99.9% of bacteria. So here you can see the difference. MIC will only inhibit the concentration of bacterial growth, whereas MBC will kill the concentration of bacterial growth. This word is important, inhibit and kill. Purpose, it identifies bacteriostatic effect on MIC, and for MBC, it identifies bactericidal effect. Method, use visual check for growth in broth or agar plate. MBC, subculturing MIC clear tubes on drug-free agar medium. Result, bacterial growth stopped here but not necessarily killed. Here, bacteria are killed, no colonies on agar plate. Clinical use, drug selection and susceptibility testing, it will be used for MIC, and for MBC, it will confirm effectiveness in serious infections. How MIC and MBC complement each other. So both tests collectively provide a full picture of antimicrobial efficacy. MIC identifies the drug's potential to stop bacterial multiplication, while MBC confirms its ability to kill. Comparing the ratio MBC per MIC helps classify drugs as bactericidal or bacteriostatic. So these insights guide treatment, especially for difficult and severe infections. So here MIC will give initial susceptibility information and MBC will confirm its killing ability. Some antibiotics have similar MIC and MBC, which is bactericidal. If MBC is less than 4 times MIC, drug may be bacteriostatic rather than bactericidal. Both are needed for comprehensive antimicrobial evaluation. Important part for tailoring treatment, especially in patients having underlying health issues, immunocompromised or immunosuppressed patients. MBC plate or subculture from MIC. So these image likely displays agar plates that have been inoculated from the MIC tubes, wells and incubated for a set of period. So plates are labeled accordingly to the corresponding drugs concentration from which the culture was taken. Growth on plates indicates live cells. The lowest concentration with no bacterial or fungal colonies corresponds to the MBC. Comparison with MIC results display which concentrations are bacteriostatic, that means inhibit but do not kill, and which are bactericidal, which will kill the organisms. So one and whole, it is image will represent minimum bactericidal concentration test. Application of MIC. MIC testing is routine in clinical microbiology laboratories to guide personalized antibiotic therapy. It aids in monitoring emerging resistance and tailoring treatment to achieve optimal outcomes. MIC data assist in dose adjustment, especially for drugs with narrow therapeutic windows, and helps public health monitor resistant trends. So application, it is used for routine clinical microbiology labs for susceptibility testing. It will facilitate antibiotic stewardships by avoiding overuse of drugs. It will support dosage adjustment based on their antimicrobial sensitivity, and it will play a role in detecting the resistant mechanisms as well, and it helps in epidemiological surveillance. MBC testing is particularly useful in severe infections or immunocompromised patients where killing of bacteria is crucial. So it is also used in evaluating disinfectants, antiseptics, and new antimicrobial compounds during the drug development. So knowing MBC helps ensure sterilization efficacy and therapeutic success in clinical scenarios. So it is important for infections where bacterial eradication is critical. It is used in drug development and quality control assays. It will determine its efficacy of disinfectants and antiseptics. It will help evaluate formulations for antimicrobial potency, and it will also assist in infection control policy development. Interpretation of MIC and MBC values. So low MIC or MBC indicates high antimicrobial potency. So the MBC per MIC ratio is greater than 4, suggests the drug is bactericidal. A higher ratio indicates bacteriostatic action. Clinical breakpoint helps categorize pathogen as susceptible, intermediate, and resistance. So clinical decisions hinge on interpreting these values alongside patients and pathogen factors. So MIC values are interpreted against set clinical breakpoints that classify bacteria as either susceptible, intermediate, or resistance. So, to an antibiotic, susceptible bacteria are likely treatable with standard doses. Intermediate, it may require higher doses or alternative drugs. Resistance bacteria likely won't respond to therapy. So, these breakpoints are regularly updated by regulatory bodies. So, MIC values are compared with these three breakpoints to categorize bacteria. Susceptible, it is highly probability of treatment success with standard dosing. Intermediate, it likely effective with increased exposure or dosage adjustment. And resistant, high likelihood of treatment failure despite increased dosage. So these clinical breakpoint will be updated according to organizations like CLSI and EU CASD. So what exactly is susceptible? A bacterium categorized as susceptible means it can be inhibited or killed by an antibiotic at drug concentrations achievable with standard dosing in the patient's body. In other words, the MIC falls below the established clinical breakpoints for that antibiotic. So this indicates a strong likelihood of treatment success using the usual dose regimen. So clinicians prefer prescribing drugs for susceptible isolates. It is important to understand that while a bacterium is susceptible, careful monitoring is still needed to avoid treatment failure or resistance development. So here microorganism is inhibited or killed by the drug at standard dosage. It is high likelihood of treatment success with the usual dosing. MIC value is below the clinical breakpoint indicating its effective therapy. Drug concentration in the body exceed the MIC at the infection site and clinicians prefer with susceptible drugs for treatment. Next is intermediate. Intermediate susceptibility means that while the MIC is higher than the susceptible breakpoint, treatment might still be effective if the antibiotic dose is increased, given more frequently, or achieves higher tissue penetration. So these category acknowledge just variability in drug exposure due to factors such as infection site or patient conditions. Using drugs classified as intermediate requires careful consideration and sometimes a dosage adjustment, offering flexibility in therapeutic options. So according to EUCAST has redefined intermediate in recent years to emphasize susceptible, increased exposure, highlighting that dosage optimization can overcome this moderate resistance. So treatment may succeed if drug exposure is increased higher than the dose or frequency. MIC is higher than susceptible but not high enough to be resistant. Useful for interaction where dosing can be optimized or drug concentrations well at infection site. It will provide flexibility in clinical decision making, and it also warns about possible reduced susceptibility. Next is resistance. Resistance indicate that the bacterium is unlikely to be inhibited or killed at achievable concentrations of the antibiotic. Even with increased dosage, the MIC is above the clinical breakpoint, signaling a high chance of clinical failure if this antibiotic is used. Resistant mechanisms include gene mutation, enzymatic drug degradation, or cellular changes that protect bacteria. It is crucial to avoid antibiotics to which bacteria are resistant and select alternative or combination therapies instead. So resistance also has infection control implication, necessitating monitoring and contaminated to prevent spread. So, in for resistant, microorganism is not inhibited or killed at achievable drug concentration. High risk of treatment failure with this antimicrobial. MIC is above the clinical breakpoint, and resistance may be due to genetic or biochemical mechanism. Alternative antibiotics or combination therapy is required. Next is factors influencing clinical interpretation of MIC. Beside MIC, factors like patient's immunity, drug pharmacokinetics, which is absorption distribution, and pharmacodynamics, which is a drug effect on bacteria, influence treatment success. So resistance mechanism and infection site also modify drug effectiveness. Thus, MIC value guide but do not solely dictate therapeutic choices. Clinical judgment remains key. So MIC result, it will do not consider patient specific factors. That means its patient's immune status or drug metabolism. All these factors will influence its clinical interpretation of MIC. Role of MIC and MBC in therapy decisions. So clinicians use MIC to select effective antibiotics and MBC to confirm bactericidal action when eradication is critical. High MIC values relative to drug levels in the body suggest treatment adjustment. So understanding the MIC or MBC relationship aids in choosing appropriate drug classes and dosing to maximize treatment efficacy and reduce resistance risk. So common microorganism tested against with MIC and MBC. Um, no, roles of MIC and MBC in therapy decision. This slide was pending. So, MIC helps select the antibiotic most likely effective against the pathogen. Though valuable, MIC and MBC tests take time, usually 1 to 2 days, which may delay treatment decisions. So, these clinicians use MIC to detect its effective antibiotic and MBC to confirm its bactericidal action when eradication is critical. So high MIC value will relate to drug levels in the body. It suggests treatment and adjustment as well. So it will guide dosing regime and duration of therapy. Common microorganism tested with MIC and MBC. Typical pathogen tested include gram-positive bacteria like Staphylococcus aureus and Streptococcus species, and gram-negative ones like Escherichia coli and Pseudomonas aeruginosa. Testing also extends to anaerobes and fungi such as Candida species. Regular MIC or MBC testing of these organisms is vital to track and manage resistant patterns. So common organism, these organisms will be tested against MIC and MBC, which include Staphylococcus aureus, Streptococcus pneumonia, E. coli, Pseudomonas aeruginosa, as well as Candida species, anaerobes, and typical bacteria. Limitations of MIC and MBC. Though valuable, MIC and MBC tests take times, which may delay treatment decisions. In vitro conditions differ from human body environments, sometimes affecting results applicability. Variability between labs and methods exists. So biofilm associated bacteria often show different susceptibilities. Tests also don't predict host immune response or drug toxicity. So it will take time up to 24 to 48 hours. In vitro condition, it may not fully mimic in vivo environment. Variability can be occur between testing methods and lab methods. In this biofilm formation, bacteria may show different susceptibilities against organism, and it will not predict host immune response or any toxicity risk. Advances and automation in MIC testing. Automation in MIC testing reduces time and increases consistency with instruments like Vitek, which is the most common one, and microscan, widely used. Gradient diffusion e-test simplifies MIC determination on agar plate, and molecular methods detects resistant genes, rapidly improve data management. Software supports interpretation and reporting, making MIC testing more accessible and reliable. Case study MIC and MBC interpretation. Consider an antibiotic with MIC 1 mg per ml and MBC 4 mg per ml against S. aureus. The MBC per MIC ratio is 4, indicating bactericidal activity. So clinically, this antibiotic is suitable at standard dosing. This example shows how MIC and MBC data should be joint considered to guide effective treatment decisions and monitor patients' response. So here is the result and comparisons or interpretation. So this image summarizes the results or shows slide by slide these comparisons to highlight the MIC and MBC points. So it illustrates the overlay of broad turbidity for MIC versus colony presence for MBC. These comparisons of both MIC and MBC allows the observer to deduce whether the tested compounds is bactericidal or only bacteriostatic at certain concentrations, a critical point for clinical or experimental microbiology. So summary of key points. MIC and MBC are fundamental tools to assess antimicrobial efficacy. Here MIC measures growth inhibition, whereas MBC measures bactericidal killing. Both of these are essential for comprehensive evaluation, guiding antibiotic selection, dosing, and stewardship efforts. So understanding their interpretation and limitations help clinicians and researchers optimize infection management. So E. coli Antibiotic susceptibility patterns of common pathogens. It will include S. aureus, E. coli, Pseudomonas, and Klebsiella. We'll go one by one. So, S. aureus species, it is highly susceptible to vancomycin, linezolid, chloramphenicol, rifampicin, and monocycline. It is showing moderate susceptibility to only gentamicin drug, and it is resistant or less susceptible to erythromycin, which is high resistant as it is reported, and some beta lactams depending on MRSA status. Next is E. coli. E. coli is a major gram-negative pathogen responsible for urinary tract infections, bloodstream infections, and several other clinical syndromes. It has developed significant resistance to commonly used antibiotics, posing challenges to effective treatment. So these colistin and carbapines, such as imipenem and meropenem, remain some of the most effective options, typically exhibiting high susceptibility rates. Tigecycline is also effective, especially in multi-drug resistant strains. However, reliance on these drugs should be cautious due to toxicity concern and the need to preserve their efficacy. Resistance is widespread to many first-line antibiotics like ampicillin and cotrimoxazole, making these less reliable for empirical therapy. Third generation cephalosporins and fluoroquinolones show high resistance rates in many regions, reflecting extensive use and selection pressure. So aminoglycoside and piperacillin have moderate activity, but resistance is increasing. Continuous surveillance of susceptibility patterns is essential to inform treatment guidelines and develop effective antibiotic programs. Next is Pseudomonas aeruginosa. It is a significant opportunistic pathogen, especially in hospital settings with notable resistance issues. So it is notorious for its ability to form biofilms and intrinsic resistant mechanism, making treatment difficult. So here colistin, and it remains highly effective against Pseudomonas aeruginosa and is often reserved for multi-drug resistant infections. Carbapenems like imipenem and meropenem still shows moderate susceptibility, but resistance is increasing partly due to gene mutation and efflux pumps. So the combination of this antibiotic shows variable effectiveness and is valuable option for less resistant strains. Aminoglycosides like likely amic acid and tobrasmin gentamicin have moderate activity, but resistance trends are alarming. So this cephalosporin, all this ceftazimine and cefuroximine and fluoroquinones are less effective due to multiple resistant mechanisms, including mutations and target enzymes. So approximately 40 to 50% of isolates will display multi-drug resistance, underscoring the critical need for susceptibility testing to guide therapy. These complex resistant landscapes means empirical treatment should be guided by local susceptibility data, and prompt identification of resistant mechanism is essential to optimize patients' outcome. Next is Klebsiella pneumonia. Klebsiella pneumonia is an important pathogen in both community-acquired and nosocomial infections, including pneumonia, urinary tract infection, and septicemia. Due to widespread antibiotic use, it has acquired resistance to multiple drugs. So here, amic acid, it will show the highest susceptibility rates, making it a reliable choice for many infections when susceptibility testing supports its use. Carbapenems like meropenem and epipenem remain effective options, particularly for severe infections, though resistance is emerging. So levofloxacin and ciprofloxacin represent moderate options, but resistance is rising, especially in hospital settings. Ceftazidimine and cefoperazone also exhibit moderate efficacy. Unfortunately, ampicillin here and amoxicillin see very high resistance rates, rendering them largely ineffective for Klebsiella infections. Other agents like Exole and cefuroxamine are avoided unless susceptibility is demonstrated. So continued monitoring and susceptibility testing are critical to guide effective therapy and combat multi-drug resistant Klebsiella strains, which are significant clinical challenges. So whole this summary, antimicrobials act as bacteriostatic, which means inhibit growth, or bactericidal, killing bacteria. It is classified by their effect on microorganism. Bacteriostatic, this bacteriostatic against inhibit the growth but not necessarily kill them, merely preventing their proliferation. In contrast, bactericidal agents actively kill bacteria, leading to a reduction in viable cell numbers. This distinction is crucial in treatment. Bacteriostatic drugs are effective when the immune system can clear the inhibited bacteria, while bactericidal agents are often preferred for severe infections, immunocompromised patients, or life-threatening conditions. MIC. So MIC is defined as the lowest concentration of an antimicrobial required to prevent visible bacterial growth in a controlled laboratory environment after overnight incubation. The MIC value essentially tells us at what minimum concentration a drug is able to stop the bacterial population from increasing. It represents inhibition, not necessarily killing, and is critical when choosing antibiotics and their dosage for therapy. MBC. The MBC is the lowest concentration of an antimicrobial which will kill 99.9% of original bacterial population. While MIC measures the ability to halt growth, MBC confirms whether the bacteria are actually killed by the drug. MBC is particularly relevant in severe infections such as endocarditis or meningitis, where persistent bacteria can be dangerous. Principle behind MIC: stops growth, MBC ensures death. So the understanding principle is that MIC determines the threshold concentration for inhibition, that means no visible turbidity, whereas MBC determines the threshold for killing, no recovery of bacteria on subculture. So in nearly all cases, MBC is equal to or greater than MIC. Methods for determining MIC: determination, broth dilution, macro and micro. Serial dilutions of antibiotics are prepared in both tubes. Macro dilution or micro titer plates, that is micro dilution. A standard bacterial inoculum is added and tubes or wells were incubated. The MIC here is the lowest concentration without visible growth. Agar dilution, the antibiotic is incorporated in varying concentrations into agar plates. Bacteria are spot inoculated, and the MIC is the lowest concentration with no visible colonies. Third is E-test stripe. E-test, this commercial plastic stripes contains a gradient of antibiotic concentration. It is placed on an agar plate swabbed with the bacteria. The MIC is red where the ellipse of bacterial growth inhibition intersects the stripes. Last is automated systems. Here instruments perform both microdilution automatically, improving efficacy and standardization for routine laboratories. Next is MBC determination. MBC determination is an extension of MIC testing. After identifying the clear non-turbid wells in an MIC assay, samples are taken and subcultured onto drug-free agar following incubation period, and the lowest concentration at which no bacterial colonies will grow in the MBC. This will confirm true bactericidal activity. Interpretation of MIC and MBC. Relationship: MIC is always less than or equal to MBC. MBC or MIC ratio, if the ratio is 4 or below, the drug is considered bactericidal. If the ratio is greater than 4, the drug is considered bacteriostatic. Application of MIC. Dosage selection: it helps determine the minimum effective concentration for patient's treatment. Detecting resistance: high MICs can signal resistance development. New drug testing: all new antimicrobials are tested for MIC to quantify